applied mathematician from Tulane, works in biofluid mechanics. And she's going to give the first of 3 lectures.
Thank you, Mike. Well, I agonized for hours over the titles of my 3 presentations, and the organizers decided not to put them up. So this is amazing simulations, perhaps. So I'll try not to disappoint you then. So what I want to do today is I want to talk about what's been motivating a lot of my work over the past many years in the biofluids of reproduction. Like Mike said, I'm an applied mathematician / computational scientist, and so I also will spend some time today looking under the hood. No, like I say, no black boxes. I want to talk a little bit about what goes into some of the beautiful computational models that you've seen. Not too much, but I just want to, I know some of you are already doing hardcore computations, and many of you are experimentalists, and you do think about computations more as a black box. So I want to just point out some of the issues that those of us who are developing numerical methods for these moving interface problems have to deal with. So today I'll talk about biofluids of reproduction and motivate some of the methodology through these problems. Tomorrow I'll spend some time talking about very low Reynolds number flows, some problems that are driven by understanding the motion of phytoplankton, and we'll talk about a very nice numerical method for the Stokes equations. And then the next day I will talk about some undulatory swimmer problems. Okay, so I will, everything that I show you is done in collaboration with different people, and I will try to remember who they are as I go through. So I see in the program that by now you're in your 4th week, and I know it's not all in biofluids, but really much of the motivation and the study of biological fluid dynamics that I think as a discipline started in the mid-50s with G.I. Taylor in Britain really is motivated by problems in reproductive biology. And you don't have to look any further to find great motivation for studying moving interface problems and fluid structure interactions than occurs in mammalian reproduction. So here's, I guess I'm here with a bunch of physicists. Maybe you need to learn your birds and bees. So sperm is introduced about here, and then they have to make their way through the cervix, where they encounter non-Newtonian cervical mucus, which is much of the lining of the uterus. They've got to make their way from the uterus through the tubular junction here to a place where there is an ovum hanging out ready for fertilization. Fertilization occurs about here in the ampular region. Okay? Once this egg is fertilized and becomes sort of an embryo here or a fertilized egg, this egg has to make its way back down through the fallopian tube. By the way, the oviduct and the fallopian tube is the same thing. The fertilized ovum has to make its way down through here and then comes back in the uterus and has to get implanted somewhere in the fundal region of the uterus. Okay? All of this is happening in a fluid environment. It could be a complex fluid environment. And on top of that, it is not a static environment. There is motion induced by the motile sperm. Okay? So there are these, you can think of these as actuated elastic fibers carrying a payload, undergoing biochemically induced motion. On top of that, both the uterus and the fallopian tubes undergo muscular contractions. So they are also causing the fluid transport. On top of that, within the fallopian tube, there are cilia. So these hair-like appendages, very much similar to sperm flagella that are undergoing, that are actuated and moving flow around. So we have an incredibly complex and interesting interaction between motile sperm, beating cilia. There is some reports of actually uterine cilia, but I'm not too sure. I don't see that too much in the literature. And so we have muscular contractions of both the uterus and the fallopian tubes. And also another interesting thing is that the fertilized ovum has no motile capabilities. So it relies on ciliary motion and on muscular contractions in order to reach the right implantation. So what are the ingredients as far as what's driving the fluid mechanics? I just said that: motile sperm, muscular contractions, and ciliary beating. So here I will tell you much more in a couple of days about the project that we're working on with Susan Suarez to try to understand the biochemistry of hyperactivated motility in the mammalian sperm. And I will tell you that I guess it was a year and a half ago, Susan Suarez, we visited her labs, Sarah Olson and another collaborator who was a postdoc at Tulane. And Susan is a sperm physiologist. She's in the veterinary school at Cornell. And she thought that it was very important for Sarah and I to see her whole operation in her lab. So we started with adorable mice, where she put in this plastic cylinder, covered it, and the mouse wasn't so happy anymore. And they took this dead mouse out, and someone who didn't want to say the word sperm yesterday? Okay.
No way.
Well, so now I like to tell this story. You know, mice testicles are internal, so they took this dead mouse, put a little incision, scooped out his 2 testicles, took one out, put it in the freezer for later, and then went over and got this, sliced open the teeny tiny testicle and got out lots of live sperm, and we looked at it right under the microscope here. And I'll show you, I'm going to talk more about this, and this is where I've become completely converted that in many of the fluid dynamic models that we look at for sperm motility and so on, the mechanics is very complicated, and it's nice to make some progress into understanding the coupling of internal mechanics and fluid dynamics. And much of the time, we, including myself, ignore biochemistry. But the moment that Susan had this collection of sperm, fresh sperm under this microscope, she added some particular chemical, I can't remember what it was, like Marisol or something. And in a split second, they went crazy. So they started swimming in circles. They somehow activated a hyperactivated pathway. So to see something, I was very moved that before my eyes, putting in some biochemical signal totally changed motility. And so I think that this is something, I should be saying this at the end, but when we're trying to work with biologists and look at modeling, there are big chunks of things that it's more comfortable to ignore, but I think that in the next class of looking at models, we have to include biochemistry. So there's my preaching for today. Okay. So you all see these animations or renditions of all of this army of sperm racing towards the egg right? Even in Woody Allen movies. But what's the deal? How many actually make it to the site of the egg? So it's, even though there are tens of millions of sperm introduced into the female reproductive tract, only 1/10 of those reach the cervix. 1/10 actually can penetrate through the cervical mucus to reach the uterus. 1/10 of those make it to the oviduct. And it's been thought that a few tens of sperm actually make it to the site of fertilization. So that's something that is pretty interesting and contrary to this idea that many of us have. So the question is, what's guiding the sperm to the egg? Now it's been known, it's been characterized for invertebrate sperm that they have to follow chemical signals because they undergo external fertilization out in ocean environments. So the eggs are released into seawater and the sperm have to make their way there. So for invertebrate sperm, there are the chemotactants in many cases have been identified. So the question is, are mammalian sperm chemotactic? I think when I wrote this slide a few years ago, since then there's been some more work, but the evidence is that there is some chemotaxis. Is that each month one side versus the other ovulates. So there were some studies done that showed that there were more sperm in the ampular region in the ovulating oviduct. Now the question remains, is it the sperm that are smart, or is it the female reproductive tract doing something to urge the sperm towards that side? So here, is this sperm chemotaxis or something else? Now, you would agree that there are few experiments on humans for this, for pretty obvious reasons. But there's been, there was a study in '97 where some female subjects allowed albumin macrospheres to be introduced into their reproductive tract, and in fact, more of those do end up in the ovulating oviduct. And so these albumin microspheres were not motile. So that's perhaps something. So the current thought is that chemotaxis perhaps does not play a role at large distances, but when the sperm finds its way close to the egg, that there perhaps are some chemical signals. I know that there's some interesting work out of Zimmer's lab at UCLA, where he's now characterizing some chemoattractants for human sperm, and I think he is, well, I think that he's working with Roman Stalker at MIT, who are looking at microfluidic experiments to study that. So I'm taking you through a gallery of interesting fluid structure interaction issues that could be served well by modeling. So here I took this off of Professor Talbot's website from UC Riverside, and here what you're seeing is the end of an oviduct. I believe it's hamster, I believe. And what here are some spores, or think of these as just particles, and you see motion. You see these moving. What you can't see here is that on the outside of this oviduct there is an array of cilia, and so the cilia are beating, and they can cause a flow that transports these spores across the oviduct. In this particular system, the ovary releases an ovum, but it's not directly released into the entrance to the oviduct, which is about here. So an ovum would have to make its way across the outside of the oviduct and turn in, as I'll show you momentarily. So here's a beautiful video from Professor Talbot's lab, showing the oocyte, which has this loosely packed viscoelastic matrix on the outside. And you see that it makes its way, unlike the spores that fall off, it makes its way across the outside of the oviduct and turns in. So it is thought that the flow field caused by the cilia is not enough to allow it to turn back in, but there has to be some mechanical interaction between the ciliary tips and the matrix of the oocyte complex. And this mechanism of cilia interacting with another elastic structure is also seen in respiratory cilia, where the respiratory cilia are thought to actually engage with the mucus that it's trying to transport. So I'm showing you this. I'm not going to show you amazing simulations of this for sure, but I'm hoping to show you that there's some fascinating systems within the biofluids of reproduction that are begging for more understanding. Yeah.
Maybe you said this, but the fluids in here are characterized, the rheology of these?
Probably not. You know, the thing is, it's nice, we all know Newtonian fluids, Navier-Stokes equations. We would like to understand viscoelastic properties. To say, you know, even within a cell or within the cervix or so on, what type of model should we use? Is it an Oldward B model? Is it a Maxwell model? It's probably something much more complicated, and that is still open for study. So I'll talk a bit today about some of the things that Mike was talking about yesterday and look at some simple complex fluid models. Okay, so actually, again, this is something about biochemistry, or the used OCCs can't make the turn and fall off. So what's going on here? What she has done is taken this oocyte complex. It went and was actually taken into the oviduct. Then she took it out again and put it on the outside of the oviduct. And so here, something changed, whether the mechanic, something maybe with the outside of the cell, maybe that was biochemical, or maybe it was mechanical or so on. But once it already went in and made that turn, once she took it out, it couldn't do it again. I have no idea. So the other, one other thing that's very interesting that just, again, comes back to classic problems in biofluids is the idea of peristaltic pumping. So moving fluid in a flexible tube by passing a wave along it. So physiologically, peristalsis occurs in many parts of the human system, in the ureter, in the kidney, and in the uterus. So one of the important issues, even if sperm motility right now for affluent humans is not all that important to have a successful fertilization, right? So there in vitro fertilization has been around for how many years now? 40 years? I don't know. 40 years. So in vitro fertilization is very successful at producing a fertilized egg in vitro. 90% of the time that's effective. What is not effective, so probably many of you know people, friends or people in your own family who've gone through this process. What is not effective is successful implantation of the embryo. Okay? So there are some agreed upon practices. It's put, a few embryos are put in a catheter, and the catheter is injected to some point in the uterus. The question is, how should that be timed, or should it be timed with the actual native propagation of waves on the uterus. And the question is, if you're inserting it with a catheter, should you, what sort of pressure should you put? So there's been some very nice work done by Elad's group in Israel, where they're looking at just simple models of the uterus as just a sinusoidal channel with a catheter here, and they're looking at what the fate of fluid markers are as they are injected with a pressure drop into this channel. And I suppose in this simple model, what they find is that the actual speed of injection is critical, and perhaps there's some insight into the timing of the introduction of these particles with the peristaltic wave. The problem with these calculations, well, it's a start, but one of the problems is that the embryo, like we saw, is not a point particle. Okay, it actually has its own, it has volume, and I think it's maybe about the 10th, and it might be about, the diameter is about a 10th of the diameter of the uterine channel.
It's critical to what? The injection speed?
Oh, okay, the delivery of that point particle towards the top of the uterus. Otherwise, it falls off. So it's critical. So if you're looking at the fate of the particle, you know more or less where you want it to go to. Okay, so the thing that they were measuring is the actual fate of the particle that's introduced, looking at its trajectory and where it's going to end up.
You said that 90% of in vitro fertilization is successful?
No, it's not successful. I'm sorry. It results in a fertilized egg. 90% or so. But that drops. But then you have to take this test tube embryo and implant it. That's when it goes down to 25–30%.
And that second stage is what you're describing?
Yeah.
Oh.
Yeah. So, there's been a whole series of papers here. So this is interesting. We see that that swimming, viscoelasticity, pumping, and so forth are all ingredients in the biofluids of reproduction, and we would even like to get some insight by looking at caricature problems. Of course, we'd like to study everything together, the coupled system of moving sperm, muscular contractions, beating cilia, biochemistry, internal force-generating mechanisms. So there's a lot to it. Okay, so the way that we're going to look at these systems is as whether we have our swimmer or a pumper, the walls of the uterus, or the swimming microorganism. We're going to think about these somehow as force generators, whether internally actuated or not. And we want to look at their interaction with a surrounding fluid, perhaps non-Newtonian if we choose a particular model. This Newtonian fluid we might be looking at very low Reynolds numbers where we can use the Stokes equations, or we might be looking at moderate to high Reynolds numbers, not in the biofluids of reproduction case, but in other applications that I'll show you. And what we would like to do is to look at this coupled system, and from understanding the physiology of the system and understanding how, due to muscular contractions or due to microtubule dynein systems, how these forces are coupled to the surrounding fluid. And from that, we would like to study a list of emergent properties. For instance, the swimming motion of the sperm flagellum, or the actual time-dependent geometry of the uterine walls. Okay, so that's a main goal. And in all of this, I guess maybe this should come towards the end, that the current challenges, and I'll show you some of the current successes, but the current challenges are really to look at multi-scale models that include biochemistry, include complex descriptions of complex fluids rather than Newtonian fluids, and full 3D models. So this guy here has nothing to do with the biofluids of reproduction, I guess, unless you're a lamprey. So one of the themes that I'll be talking about in the next few days is trying to understand how the internal force-generating mechanisms couple with a surrounding fluid. And so I'll get to this, looking at the internal structure of sperm and cilia in a bit. And one of the other projects that we'll be looking at later on in the week is a swimming vertebrate or lamprey. Okay. I have a student who's supposed to be finishing his PhD thesis by Friday. I'm going to have a draft where he's looking at the actual mechanics of uterine wall force generation. So that I put that up there to remind me to send me an email. So this is a very many of you have seen this slide before. This series of micrographs from Susan Suarez's lab. I don't think she realized that this was going to be a favorite of applied mathematicians and physicists. And it surely demonstrates the idea of fluid structure interactions. So I want you to look at this panel and this panel. Okay? So here, these are bull sperm, and here are micrographs of superimposed of a bull sperm swimming in a Newtonian fluid, and you see that there's this very nice symmetric beat pattern with larger amplitude at the tail. Okay. What I want you to look at over here is the same bull sperm or its cousin, I'm not sure, put now instead in a viscoelastic fluid, in a non-Newtonian fluid. And you see that the waveform is distinctly different. You see it's mostly straight over here with some wiggling or some figure 8 pattern at the end. So this is just is a demonstration that many of us have used this to demonstrate that solving, to understand sperm motility, solving the kinematic problem where you input a waveform and you look at the surrounding flow field and swimming mechanics is not enough. There's actually interaction between the internal force-generating mechanisms and the surrounding fluid. You change the surrounding fluid, your beat form looks different. Okay, and so that should be, this is like a religious slide that's motivating on what you're looking at over here, just to tell you, is what's characteristic of hyperactivated motility. And hyperactivated motility occurs later on in the reproductive tract when these sperm are hanging around. So I didn't tell you that when the sperm actually make it to the oviduct, most of them who are there, they get caught in mucosal folds in the oviduct, and they hang around there until it's ovulation time, a few days. And at some point, and there's some biochemical cascade that causes calcium influx into the flagellum that changes their beat pattern from this symmetric beat pattern to something asymmetric like this. And it is thought that this wild asymmetric beat pattern enables them to generate enough force to free themselves from the mucosal folds. Okay, and it is also thought that this beat pattern enables them to penetrate the outer layer of the cumulus complex.
I'm probably not alone in wondering how human sperm compares in size and
Let's see. So I can't think of both. Mouse sperm are a lot longer. They're all pretty much the same. What differs is, I'll talk about the internal flagellar structure in a bit. What differs is perhaps some of the outer sheaths that's on the flagellum. So in terms of size, mammalian sperm, whether maybe human sperm, it's leaving me right now. It's about 30 microns. Stephen?
Yeah, something like
It's about 30
Good.
microns.
that. Roughly, the smaller you are, the bigger your sperm are.
Yeah.
Like my sperm are bigger than human sperm, and
Really?
fruit fly sperm are bigger than
Yeah.
mouse sperm.
My sperm is about 120 microns long. So this is bull sperm, and they have a nice kind of symmetric head. What I showed the mouse sperm before, the mice are very cooperative. They've got a sickle-shaped head. They've got a hook on their head, which they're cooperative in a couple of ways. It's been shown that for some species of mice, when these sperm are moving towards the egg, they form trains. So they hook this hook shape on their head enables other heads to join, and so there's a whole bundle of sperm, and they're called. This is cooperativity in the biological sense, because the front runner loses the ability to fertilize the egg, but the guys that he's pulling along are gaining energetic advantage because they're being pulled up to the egg, and they don't lose that ability. So that's called sperm cooperativity. And so that hook form for mice. And so
Why does the first guy lose the ability?
I don't exactly remember why, but it undergoes the axoneme reaction prematurely. There's some chemical reaction on the cell body that needs to happen in order to penetrate the egg. And so I think that's true.
It's not fair.
Well, see, here's the thing. Unlike humans, mice have many partners. And so there's some thought that this type of cooperativity, you want one of your guys' sperms to be the one that fertilizes the egg instead of somebody else's. So there's a whole bunch of literature on cooperativity in sperm. Great, good, finally we get to some equations. So what describes all of this? Okay, so we all know the Navier-Stokes equations here. Mass times acceleration equal applied forces, and incompressibility. But now what I'm going to do, and most of the simulations that I show you are, I'm going to take what I call an immersed boundary framework. Okay, so rather than thinking of my moving uterine walls, let's say, as being boundaries along which we will have to specify boundary conditions, I am going to imagine that whatever actuated structure like a sperm flagellum or like the walls of the uterus, that these are immersed fibers within a bigger box of fluid. And I'm going to think about these fibers or these immersed elastic structures as generating force on the surrounding fluid. So my goal is, like I said, my goal is to look at numerical methods to capture moving interface problems coupled with the surrounding fluid. So I guess I can use this blackboard. So I'm going to have a box. Probably I'll stick to 2D for the description. And within that, I'm going to have some moving structure. So to start with, I want us to think about, let's just have a 2D box here. A 2D fluid domain within which we have something that is generating force. And in 3D, we might have something that's generating force that has more structure to it. So the way that I'm going to think about it is that this structure, think about it as just a flagellum like that, but I'm thinking that this structure is built out of K filaments. And each of these filaments, which I'm calling XK, my color coding is almost consistent. that there is some structure, a Lagrangian structure, xk, where there's my k filament, and in fact I only have one here, so I'm calling that x. And that's what I've written in red here. And this structure here is going to be time dependent, and it's spatially dependent on, say, something like arc length. So we're going to look at this structure as it moves through a fluid domain. So the Navier-Stokes equations here, I'm writing them in Eulerian form in the sense that here u, my velocity, is a function of x and t for a fixed Eulerian point in space and at time. But my fibers, which I've written in red here with superscript k if I have my k fiber, this is free to move throughout the domain. And what you see here is this. What does the force do to the, on the fluid, due to the k fiber? So imagine that this fiber is a series of Hookean springs. Just think of it as a rubber band or a series of springs. At each point on that fiber, I have some sort of spring-like force. That's the easiest way to think of it. And this force on the fiber at the position x and s and t is this red force here. Think of this as something that is living on the fiber. And the way that we communicate it to the surrounding fluid is like this, where here, this is a Dirac delta function. So here, the force on a fluid point at position little x here, black x, is only going to be non-zero if our fluid point coincides precisely with the point of the fiber. Otherwise, it would be zero. Okay, now, this description is a way of relating fiber forces to forces on the fluid. Now, this is a singular force that, in the sense that it is supported only along, if we're in 2D, the force is supported on the fluid only along fluid points that coincide precisely with that curve. And so you can see that numerically we'll have to do something to mollify that. Okay. But right now there's nothing numerical in what I'm writing here. I have the Navier-Stokes equation, and they are going, the elastic actuated fiber that we have is going to be included in this framework by acting as a source of time-dependent force. So this integral equation represents communication between forces that are on the structure to forces that are on the fluid. In order to close the system and to have the feedback from the fluid onto the fiber, we satisfy the no-slip condition that says that within this, the velocity of a material point on the fiber has to coincide with the fluid velocity evaluated at that fiber point. Okay? And so we can write this in a way in parallel to this with the Dirac delta function as such, so that the velocity on the fiber will be just pick out the fluid velocity there.
Does this concern momentum?
Yes. It depends on the type of forces that you put. Okay? So one can, and I'll talk about that. Okay? So this here is a continuum description of a coupled elastic fluid dynamic system in a Newtonian case. We could do the same thing. We can just get rid of this and put a 0, and we have the Stokes equations. So I'm going to, let's think about what these forces are in the simplest case. So here is a mathematician building a sperm. Okay. I did it a while ago. So suppose you want to You don't want to talk to biologists. You want to just think about what forces do you need in order to have an elastic filament that's actuated in a fluid. Well, you want, so think about, what I showed you before was a continuum description. What I'm talking about now is a discretization of that. So first, let's imagine that we have this, here's my red X here, a cell body and a filament. And let's imagine that we're going to build this, discretize this filament out of a whole bunch of points. So the first thing is we want this to maintain material integrity, so we don't want the points to ever be able to fly apart, so we have to connect them. So we connect them by linear springs. Okay? We could think about that in terms of an energy, and think about that if we have two successive points on my sperm tail, we want these to have a rest length of delta S, and we'll endow some stiffness S1 to this tensile force. Okay? So that's, I'm writing down an energy here. Great, so that would be just for a passive filament and a passive cell body. On top of that now, we want to actuate this filament. So we could do this in a whole bunch of different ways, but if you think about it, I mean, you've seen talks on the three-link swimmers and so on. You want to actuate the bending angles at the links. So one way of doing this is to write down a bending energy, and the form that I'm writing it down here is the cross product between these successive links. So the cross product of two links in 2D is, you know, a scalar, and it's just the angle. It's basically the angle between these links. And if we put in some driving function or some preferred curvature, this is related to the curvature up to some scalar multiple. So if we specify a time-dependent driving function and write down this energy, we can then compute the forces along the structure just by differentiating this energy. And that's what we do. We can write this in a continuum way. Perhaps I should have looked at this first. If we want, when we think about wanting to model just some filament by a bending energy where we're specifying a time-dependent curvature, and by some tensile energy where we're specifying the arc length. And inherent in this formulation, and inherent in most immersed boundary calculations, are these stiffness constants, which tell you, you could think of them in two ways. You could think of them as numerical parameters that if you crank them up high enough, you will get a desired kinematics from putting in that prescribed curvature. Or you could think of them, the better way to think of them is as material quantities that tell you something about the passive elastic properties of your structure. Okay. So this is a classic movie from 1994 where we put two such creatures in the domain. And I think that I had to digitize this from a VHS tape, which was better than 8mm tapes, which is how they started. And you know, this looks like a cartoon, but it still is pretty impressive, and I will tell you why.
Who's moving who there? I think, if I understand correctly, you're getting this little sperm cartoon to move, and then looking at the fluid, the way it's responding to that? Is that right? Or is it the other way around?
Who's moving who? It's a coupled system. So, each of these guys are endowed with tensile and bending forces. Each one of those, forget the cell bodies. The cell bodies, of course, have no bending, no actuation for bending. But each tail is given the same stiffness constant and is told, here is your preferred curvature. We would like there's the energy function. you're going to exert a force based upon this energy function. Now, if the only force was the gradient of this energy function, and there was no fluid anywhere, the motion would minimize the energy and we would have the precise shape that we think we're prescribing. In fact, this is coupled completely with the fluid dynamics. So we have a force coming from this energy that's being spread to the fluid. The fluid then is responding in its velocity field, and we are moving these structures in that velocity field. So these two structures are being actuated. I should also say that we are representing elastic walls here with they're pretty rigid. They're also elastic structures. But what's the beauty of this framework, this immersed boundary framework, is that each one of the structures is just adding to the right-hand side. It's just adding to the surrounding, to the external force. So then we could solve the fluid equations in one fell swoop without worrying about moving boundaries. It's just a force field. So what's coming out of this calculation here, what is impressive is that you see that the relative motion of the sperm is such that they phase lock almost immediately. Okay, this is not something that is input to the calculations. This is an emergent property. We see synchronization. We see the interaction because of the viscosity of the fluid. It immediately phase lock. What you also see is that there's interaction with the walls here, and that I'm not telling you what my specified curvature was and what my preferred amplitude is, but they flatten out because there is viscous resistance from the walls. Okay, so we could have done, we could have put 2 sperm, 6 sperm, whatever. This is 94, which I guess is a few years ago by now. But while these are very interesting fluid calculation, there's a number of problems with them. It's 2D. We'd love to do 3D. This is really a swimming sheet, and the cell body is a cylinder. Okay. And also, there's no physiology in here. This is, like I said, an applied mathematician's creation of sperm swimmer. Okay. So I'm telling you at the outset what the drawbacks are, but still what we have are full fluid structure interaction calculations where we see, even in the simple case, synchronization. Yeah.
Did you run any calculations where you started the sperm farther apart and if it phase locking there, or whether they would get closer to one another? Or?
Back then, I think yes, we looked, and we're looking at other simulations right now. We're looking in a complex fluid, so the phase locking is definitely enhanced when the distance is close. So, looking at those systematic studies for sure. We looked at that time this model of a single actuated swimmer near a wall, and in fact, it's in this 2D case, the wall acts as an attractor. Okay, and I think, this is 1994. This is when it wasn't in vogue to study low Reynolds number of swimmers, but since then there's been there's been a lot of work done on looking at swimmers near walls and 3D and so on. Okay, yeah?
So does the picture change if you make it non-equilibrium by including a noise?
I'm sorry, I can't hear that.
Does the picture change? Does this change if you make this thing non-equilibrium by including a noise which is not in fluctuation dissipation with the...
I've not tried that. So I want to get past 1994. Okay. But no, that's good. So with this capability here, there's a lot of really. I've left this now a lot of years ago. But even within this structure, there's a lot of questions that could be asked. See, at the time, physicists were not interested in this problem. It was the biologists and the asymptotic people from Britain who said, this isn't 3D, and the biologists want much more realism. But I think even within this system, there are a lot of what-if questions with this very simple system. but I'll show you how we moved on since then. Okay, so when I look back at that system of equations, the Navier-Stokes with forcing, here's my framework. What's happening here? We have some structure that it's still in red, but my superscript is now a subscript. From that configuration, we would like to see what the forces that are, or what forces it generates. This little arrow here takes a lot of my time because this is where all the interesting physiology and biology comes in. What are the, given the structure, what are the forces that it's generating? This might be coming from biochemistry, this might be coming from internal motors, muscle models, and so on. But for now, let's just think, we have a configuration, it's generating force. In the Navier-Stokes realm, what we do is we're going to take a finite difference approximation. So we're going to take this box and overlay it with a grid. Now we can do this in many different ways. We could even do finite element description, finite volume, and so on. But imagine that we're going to discretize our fluid domain. And somehow we've got to get forces that live on this Lagrangian structure and communicate that to the surrounding grid. We can't use the Dirac delta function, but we use blob functions or discrete delta functions and so on. And I'll talk more about that tomorrow. But once the presence of this structure is just represented by contributions to the force field on the grid, we've got to solve the Navier-Stokes equations without any special boundary conditions. We can think of a big periodic box, if that's what, a big box with periodic boundary conditions, if that makes sense. So this part here is very fast, and in 3D it's very fast as well. And so from that, then, we have to go back and take the values that we computed for the fluid velocity and interpolate that grid velocity onto the filaments and update the position. So, let's see. I think that I think in the interest of time, if I have some more time tomorrow, I'll go through this. I was going to get into some more of the details of what goes into here and what goes into here, but I think I'm going to save that for tomorrow. But suffice it to say that for those of us interested in using this as a numerical, as a black box, that is possible. There's some great software that has been written by Boyce Griffiths at NYU Cardiology, where he has an adaptive mesh version of this, and that's what we use for our Lamprey project. But in fact, I contend that in these complicated problems, one ought to never look at these interface problems as a black box. You should have someone on your team who knows what's going on in there. So what makes all of this complicated? What would we like to do faster? What I've written down here is an explicit time-stepping scheme. You have a configuration, you compute the forces, you apply those forces, get out the velocity, and update. So for those of you who have any experience in numerical methods, explicit time-stepping, when you apply a force, is a recipe for disaster. Nevertheless, if you do implicit time-stepping, that's really hard. So there's a give and take. You might have to, and a lot of what I show you, we are doing explicit time stepping. Your time step is very restricted. So here is just some amazing simulations now. Just some pictures using this framework. I should say, what I'm going to talk about tomorrow is this transition here, where suppose that we don't have the Navier-Stokes equations, where the Reynolds numbers look small enough that we could make the Stokes flow assumption. There the equations are linear. You have a delta function forcing the Stokes equations. You will get the classical Stokes-Lit solution, which I will derive for you. Numerically, the Stokes-Lit solution is not so great because it's singular where you apply a force. So if you're applying a force somewhere and then you want the velocity at the point where you apply the force, infinity is not going to be so good. So there are ways around this, and so it's a very nice numerical method that I'll show you that works within an immersed boundary framework, but for Stokes law. Okay. So there's this calculation is actually using a particle method in Stokes law. Here's an old movie, just to show you. Okay, so in the time that I have left, I want to show you two contributions since 1994, where we're looking more at what's happening internally to actuate a sperm flagellum and cilia, and then I'll show you some success we've had in looking at these simple models, not in a Newtonian fluid, but in a simple complex fluid. So, did anybody in the workshop talk about the 9+2 axoneme?
Very briefly.
Okay, good. So I'll speak about it briefly as well. The amazing thing, if you take a sperm flagellum or a cilia in your respiratory tract, its internal structure is preserved. Even if you look at parasites that live in the hindgut of cockroaches, this is what their flagellum looks like. There are some variants, but basically this is preserved. Somebody say something? Actually, I am not supporting this, but this is one of the structures that the intelligent design people use to support their claims.
Well, it could well be used to support evolution, because if it evolved once and if it's good, it will always be there.
That's right, so we can use it too. There are some variants. Okay, so the 9+2 structure, in a eukaryotic axoneme, there are 9 microtubule doublets that are on the outside, 2 on the inside. There's lots of protein structures there. There are links between. There are lots of protein structures, yes. The functioning of all of these protein structures to this day is still not agreed upon. You might hear conflicting points of view about Nexin links. So there are these barrel hoops that hold the whole bouquet together that seem to be not non-dynamic. So other people will tell you that these barrel hoops are also dynamic. What is agreed upon and what's very important for motility are the dyneme motors here. So these dynemes act on the neighboring microtubules. They undergo a conformational change that's ATP driven, and what happens is they grab onto the neighboring microtubule. When they undergo a conformational change, the 2 microtubules slide relative to each other. Now, unlike the lamprey spinal cord and other things, there is no central pattern generator that's telling each of these individual dynemes, you activate, you next, you next. So somehow there is coordinated activation of individual motors, both in space and in time, that gives the beautiful, regular beat patterns that we see in a beating cilium, power recovery stroke, and the sperm flagellum. And like I say here, this is still true, even though this slide is a few years old. The precise nature of the spatial and temporal control mechanisms is still not understood. There are hypotheses about how this dyneme is activated. It is understood that it's calcium. Calcium is very important. It is understood that it's ATP driven. But when does one dyneme activate and then its neighbor activate? So Charles Lindman has proposed a geometric clutch hypothesis that says that the activation depends upon the forces pulling microtubules together, calls it the T-force. Charles Brokaw has been studying eukaryotic axonemes since the 60s at Caltech, contends that it's a curvature control mechanism, so that somehow this diamine feels the local curvature and is more readily able to attach depending upon the geometry. And the two hypotheses that I just spoke about are at times analogous, and there are other ones that talk about the activation depending upon the velocity of sliding. So this is what I'm going to show you is joint work with Bob Dylan, who is also an applied mathematician at Washington State. His name is spelled differently. And so what we're seeing is, but my Bob Dylan also plays guitar. What we seek to do is to use the coupled fluid immersed structure system as a test bed to look at different theories of diamine activation and see what the resulting macroscopic beat forms are and fluid properties. Okay. So here's the open question: how are these diamines regulated? Here's the axoneme. Now what we would love to do is to put in the entire structure, and I probably proposed this to NSF maybe years ago until I realized that it's not possible to do all together. We would love to put in the full 3D structure, all of the right passive elastic properties, some are measured, some are not, put in biochemistry and so forth, and let this roll. But in fact, you can't do everything. So I took a slide from Chris Johnson. You want to model the bull, but you have to, we're physicists and applied mathematicians, so you get the essence of the bull. And that's what here in one series of models that we're going to look at, we'll try to do. So we started this about a while ago, and we're still using this framework in some sense. We're looking at a simple model where we have two microtubules. Okay? Now microtubules, what you might have heard about, they're fairly bending resistant. They have some compliance, but they're bending resistant. What we're going to do here, I don't have this color-coded, but here we're going to build a single microtubule out of two filaments with cross supports. So, think Hookean springs. So I don't want to put in any bending forces here. I want to get bending rigidity from opposing families of springs and cross supports. So here's microtubule 1, here's microtubule 2, and between them I'm going to have forces, and I'm going to have dynamic springs. So my diamine motors are these diagonal links here. Now imagine that these diamine links here are springs. They're really nonlinear springs, but imagine that they're springs with rest length 0. Okay? Now imagine that these microtubules were not tied down at the cell body. And imagine that all these diamines were activated, meaning their spring forces are working. They want to contract to be rest length 0. Which way would these microtubules, what would happen? Do they slide relative to each other? We also have forces keeping them a certain distance apart. So Pecco went, right. So this would want to go up and this would want to go down. That's what's happening inside the actual axoneme. The microtubules are sliding, but that sliding is converted to bending. It's just like if you took a paperback book that's bound and you tried to slide the pages past each other, it would bend because it's stuck at the binding. Okay? So here, what we're going to do in this model, like I said, we'll build the microtubules out of rigid links. We're going to put in the other protein structures as forces between these things. And then the dynein motors will be also represented as forces, and these forces are going to, think about them as dynamic springs that as the microtubules slide relative to each other, they can attach, detach, and reattach. So these springs don't always connect point A to point B, they can change their topology dynamically as the microtubules slide. Okay? So I just said that. So the dynein arms or the dynein motors can reassess they can assess the sliding, they can be stretched to a certain point, they could attach and reattach. Okay? It is fairly established that during a power stroke of a cilium, so now I'm going to look not at a sperm beating, but at a cilium. During a power stroke, all the dyneins are activated in that direction. During a recovery stroke, and we all know about reversibility and Stokes flow, you can't have power stroke and move fluid. During a recovery stroke, there seems to be a wave of activation of dyneins bending in the opposite direction. So here is, again, an oldie but goodie movie where we put this in, I think this is still a very low random number, Navier Stokes fluid. So I want to point out to you what we have here. You can see this cilium being made up of two microtubules. Right? What you don't see are the individual dynein motors. There's probably 1,000 of them between the microtubules. Let me tell you what's input into this simulation and what is coming out as an emergent property. What is input is the viscosity of the surrounding fluid. The surrounding fluid is indeed Newtonian. What is input is the passive elastic properties of the microtubules in terms of the stiffness of the cross springs and so on. And we've characterized the actual bend modulus and made sure that when we put in these spring stiffnesses, that gives us something that is actually a cilium and not glass or something like that. So what's being input? And then we have two families of dyneins that live between these two microtubules. Dyneins that could act in one direction and dyneins that could act in the other. In the real axoneme, dyneins are polar, they can only push in one direction. But the real axoneme is 3D. And so when it bends in one direction, dyneins on one side are activated on one pair of microtubules, and on the other, these dyneins are activated. So what we're doing is compressing our 3D structure of microtubules into a plane. And so we're letting dyneins who act in different directions share the same space. So what's happening here is during the power stroke, all the dyneins are activated, meaning all these springs at rest length 0 are contracting. They're all activated, we get this bending, and when do we stop the bending? We stop the bending when the shear between the microtubules, or the distance when the tips have slid, exceeds a threshold. So we put in that threshold. When that happens, and we say we're in the recovery stroke, we put a wave of activation of dyneins from the base to the tip, and we activate all the dyneins up until the point of maximum curvature. So this is a geometric cue. We are not saying that we are not specifying the speed of activation of the dyneins. This is all coming from the geometry. So we're not saying that this is precisely how a cilium beats, but what we are not specifying is that we're not specifying the kinematics of the waveform at all. We're not specifying the temporal dynamics. We don't know what the time course of a beat is going to be. All of that is coming from the geometry. So, and those little stars are just fluid markers to give you an indication.
So would this be consistent both with the geometric clutch model and the curvature model, or
This is the curvature control model. what we are trying to do is work with Charlie Lindemann to put in, he's got some activation code based upon the local force that the dyneins are imposing, but we haven't done that yet. Okay, so now here, this is more recent, a few years ago, we took the same cilia and put his brother in. And one of them is in a power stroke, one's in a recovery stroke, and you see that very quickly they synchronize. Okay, so here is hydrodynamic cooperativity or synchronization, or however you want to call it. We're looking at two neighboring cilia.
So are there controlled experiments or biological experiments to compare these kind of dynamics
It's
to?
very difficult to, maybe you could do this. It's very difficult to isolate two cilia. There are ways of putting in carpets of cilia and so on, but what I'm showing you is just two. It is known that two cilia close together do quickly entrain. But when you have a full array of cilia, you get metachronal waves. So I don't have a reading to put up here, but we do have we have periodic conditions. So we put in like 4 or 5, and we see that we get some kind of, not perfect synchrony, but a wave coming across. So this is a very simple model of flagellum, but the fluid dynamics is accurate. So what I think you can see one of the goals is, I think in a lot of these biofluid simulations, it's not just that computing the flow around a flagellum. Let's put in the kinematics, let's see what the flow is like. That could be a hard question, but there's much more interesting things to look at, because getting the flow correct is getting the whole system correct. So getting the flow correct is not the ultimate goal of this. We want to put in the correct physics. We want to couple this with an incompressible fluid, then we do get the flow correctly. But maybe that's not what we're focusing on. So what else do I want to show you? So here's 3 cilia. What we're also doing right now, and this is starting to look at complex fluids, in the respiratory tract there's a layer of mucus that gets transported. So one avenue that our group has taken in looking at complex fluids is to take a discrete microscale representation where we represent this viscoelastic layer as a bunch of points and springs, again, as an immersed structure on the surrounding fluid. And here's the same simulation where this green mucus layer are just fluid markers. So here there's elastic forces being imparted. Here these are just markers. So what we have now, I didn't upload a movie, but I can show you somewhere. We've got the cilia actually mechanically attaching and detaching from the mucus layer. Okay. So sperm as well. To do this, we do the same thing, but rather than having a power and recovery stroke, along the sperm flagellum, some dyneins are acting in one direction and some on the other. So here we've implemented a curvature control algorithm, and just show you here we take the same, I'm not going over what that curvature control algorithm is. Suffice it to say, we put in a threshold that if the curvature is above that threshold, then we activate the dynein, and if it's below that threshold, we don't. And we keep testing the local geometry. But we take the same machine, this guy looks like a lollipop, and we put it in a fluid of a certain viscosity and 10 times smaller viscosity. And what you see here are, you see this cartoon of the sperm, but what you're seeing is that the waveform, we're changing one parameter in the Navier-Stokes solver. viscosity, the waveform and the swimming dynamics is completely different. So what we're doing is we're comparing these. We're in the process of writing up a paper now where what we're doing is comparing these dynamics within the Newtonian case to experiments. And what's done in experiments, yes, there are viscosity changes. This was done in the 60s, and when you change the viscosity, it is shown that the wavelengths actually decrease. So when the viscosity increases, the amplitude decreases and the wavelengths decrease, which qualitatively we're getting here. There are lots of experiments that are done with sperm by looking at, they're called ATP caged experiments. So where you manage to suck out all the ATP, and so they get frozen. And then you have the sperm with this frozen waveform, and then you could put in some more ATP, and it moves on again. So what we're trying to reconcile is how in the curvature control models of Brocaw, where we put in a threshold which dictates activation or not, how does that correspond to these ATP experiments? So how does lowering the ATP correspond in our model to changing the threshold concentration? So we're trying to reconcile all of that.
So question, how long wall clock time does it take to integrate these simulations for a couple of beats?
This, 2D, a day.
Okay. For a couple oscillations of the...
These are 2D. So I said I was going to look under the hood, and I'm not looking under the hood here. These are very stiff calculations. There's a lot, you just see two filaments here. But between each of these filaments, there are motors. And the motors are examining the curvature at each time. And the microtubules, you see two filaments. The microtubules themselves are filaments with cross struts that are rigid springs. So it's an inherently stiff problem. What we are doing here that's not smart is that we are using an explicit calculation. We have our configuration, we compute the forces, spread the forces, and move on. It would be much better to do an implicit calculation. Well, if somebody wants to write the code for an implicit calculation, there are groups who are doing it. What does this mean? And you know what, I'm going to talk tomorrow, I'm going to finish up with the continuous viscoelastic. But I keep saying this, and I think that, let's say for me this is an undergraduate calculus class. Who knows what the solution to this equation is? Here is a differential equation with a very fast time. Let's imagine that lambda is okay. So lambda is 1,000. What you have here, your solution is rapidly decaying. Right? So any numerical method that you put for this, with lambda is a very big number, any numerical method to integrate this differential equation, you want to die out to 0 fast. And you don't really want to use a tiny, tiny time step in order to get that. Okay? So look, if you use a simple forward Euler, where you say yn, so let's say yn is about your solution at time level Tn. So if you use a forward Euler, y at n plus 1 minus yn over your time step should be equal to minus lambda times yn. Right, this is, you're integrating y prime equal f of y. You put in your y at your current time step here. Right? So you get yn plus 1 to 1 minus lambda delta t times ym, right? This is called your amplification factor. The cost, yn plus 1 then is this 1 minus lambda delta t to the nth power times y0. Okay? I know that some of you probably haven't thought about this in a long time. But look at this. In order for this, you want this, does this go to 0? In order to have that going to 0, you have a restriction on your time step. Right? And suppose that, let's just take lambda equal 1,000. That's a really quick decay. What you get then is that you need 1 minus 1,000 delta t for that to give something that goes to 0. This in absolute value has to be less than 1, which means that not an absolute value has to be between that and minus 1. Okay. So what do you get in the case of what do you get? This is always less than 1 because this is negative. But you get, what's the restriction on your time step? Can someone do the arithmetic for me? You get, put this over, get a minus 2 here, and then when you multiply both sides of an inequality by a negative, it changes direction. So you get, what do you get here? Help me. 2 over 1,000. Right? So what happens is, the stiffer your problem is, the worse your time step restriction is. If your time step is not small enough, then this here, your amplification factor is negative. So in fact, your y-ends, you get stuff like that. If you're clever, however, and you put in here, what I'm saying is implicit. I know many of you think about this all the time, and many of you, your experimentalists, haven't thought about this in a million years. If you evaluate your forcing at the next time step, or in this case it's really easy because it's linear. If you evaluate it at your next time step, and you do the arithmetic, you get 1 over 1 plus lambda delta t, and that's always less than 1. So your integration here will always go to 0, and you don't have instability. So that, even in this ridiculously simple example, y prime equal lambda y, or y prime equal f of y, what you really want to do when you're integrating your equations is to put here not y at your current time step, but what y is at the end of your time step. What I showed you just before, that was easy, it's linear. But now you go over to the PDE case, and all hell breaks loose because maybe you could do it. You want to put in your configuration of these elastic structures where they would be at the end of the time step, but of course that involves the velocity at the end of the time step and these integrals against delta functions and so on. So there have been, there are some implicit, semi-implicit methods, but really the linear algebra is out of control. But there are groups who are doing that. So what I hope that I've left you with today, with my simple numerical methods, one less than stability, is that when Dan asked the question, how long do these simulations take? They are, we're getting a lot for a lot of bang for our buck here in a sense, because these are solving the full fluid equations. We have internally actuated systems. We can change stiffnesses and so on. But it is an explicit forward Euler-esque calculation. We would like to do this much faster, but it takes, it would take more algorithm development to do that. And finally, so the good news about this is that even in this simple model, we are able to explore different types of theories where we actually have the curvature, the local curvature information. And one thing that we're starting to do is actually overlay calcium dynamics in biochemistry on that flagellum and have the forces that the dyneins exert actually be a function of the local calcium dynamics. I'll end with this, and this will set me up in a good place for tomorrow. And this is work of Bob Dylan and his student, trying to look at what happens when you have one of these actuated sperm moving not in a Newtonian fluid, but in a complex fluid. And in a very exuberant way, Bob put a background mesh overlaid on the Navier-Stokes mesh, where each of these are springs that have their own type of properties. I think these are a bunch of little Maxwell elements. I think he's done it with Jeffrey elements. And I'm not going to give you the full length of the story here, but you can see that this is interesting. You can see that there's this fibrous mesh being transported along. And so what Bob and his student are in the process of doing right now is to reconcile a mesh-like model like this with an Old-Rigby system. So I will continue telling you about viscoelastic swimmers tomorrow.
Questions for Lisa?
Very interesting. You mentioned the effect that the behavior of these things had when your colleague put some biochemical additive in there. What would be the next step if you really wanted to mimic that? What kinds of things would you want to know about what was added to there? And did it change just the speed, or were there maybe some qualitatively different things that might open up new horizons?
If I can't find this in a split second, I will.
I can wait.
Lord, I don't like when you hook up a computer with what happens is that it opens up certain channels on. Okay, so what happens is they don't just swim faster; they swim in circles. Okay, so they undergo this hyperactivated motility, where it, instead of having a symmetric beat, the tail whips, and they just go around in circles. This is characteristic of hyperactivation, and it is known that calcium channels along the principal piece of the tail open up, calcium rushes in, and stuff happens.
Okay.
From there, we don't really, there's been progress in understanding the biochemical cascade, but how that's translated to the motor mechanisms is something we're
But your work is heading a little bit in that direction.
Yeah,
this
and I'll talk
calcium
about
thing.
that in a couple of days.
Thank you.
So, when you showed Susan Suarez's, the famous picture that everybody knows, right? So, the one in the viscoelastic fluid, it looked like most of the flagella is straight and the motion is concentrated at the end.
That's right, because that's because of the stiffness along the mammalian sperm is not uniform. Because there are these outer dense fibers that are thicker from the base down, so the stiffness tapers.
And so in this simulation, there's no taper.
No, these sperm could be whatever you like them to be, but we think of them as invertebrates, which have no outer dense fibers. Right. So that's one thing we've not, in this model with the dyneins, we haven't tried. Because then so we see with different viscosity we change the shape, but it's not concentrated at the end.
And so I'm just, because when you put it in the non-Newtonian fluid, you don't see the transition.
No, you don't. Not in no. You see changes. I'm just showing you a vignette. I know Bob and his student have been doing a bunch of runs, but I think you really need the stiffness tapering off. I don't know why he hasn't done that yet.
It's so close.
So how does this immersed boundary framework cope with having bodies that are very close to each other? Because that becomes very problematic in sort of other methods also as well.
Yeah, I mean, in other methods that have that involve Stokes flow, like bundling of flagella, people put in repulsive forces, that kind of thing. You could, of course, when things get close together, you need resolution between them. So if you're willing to put refined mesh in between the structures, you can get around that. So I didn't talk about this today, and I'll show some things, but this is where adaptive mesh really helps. Because if you want to look at something in a big tank and you want things to get close together, you want to put more resolution close by, but you don't want to refine your entire tank. And so I reluctantly use, from my lamprey calculations and some other things, Boyce and I are good friends, I reluctantly use the adaptive mesh refined code because I need it. But I think the beauty of the immersed boundary method framework, at least in the finite difference scheme, is that once you spread your forces, you have a regular grid and you could use FFTs or other things to solve it. Once you go down the rat hole of adaptive mesh refinement, then you have to be smarter. It's good that someone else is.